All right, why don't we get started, everyone? Well, hello and good day to all the attendees and speakers. We're looking forward to a great series of talks during this Arima Neuroscience webinar. We're extremely lucky and pleased to be joined by 4 exceptional speakers. Thank you so very much to Anthony, Schahram, Jon, and Dan, and to all the attendees for being here. My name is Joe Speidel. I'm the commercial director at Arima-Genomics, and I'll be your host. Here's the structure. I'll introduce the speaker while he or she is preparing the screen. All audience members will be muted, but you'll have access to a Q&A box once the talk is underway. Please note your questions here at any time. After each talk, we'll take about 5 minutes and ask the speaker to address some of the questions from the box. If we don't get to your specific question, our team will follow up with you afterwards. All right, well, that's the nuts and bolts. So let's get right to it. And our first speaker is Arima-Genomics' very own Senior VP of Science, Dr. Anthony Schmitt. Anthony is the lead developer of the extremely impactful Arima-HiC kit, and he's the lead scientist behind the Arima-HiChiP and Capture-Hic solutions. He's an incredibly knowledgeable and valuable contributor to Arima's success, and we're very thankful for him. So Anthony, welcome.
Thank you very much, Joe. Just a quick sound check. Can everybody hear me okay? Joe, can you hear me?
Yes, sir. We got you.
Fantastic. All right. Yeah, so the title here, Discovery of Genomic Interactions and Gene Regulatory Mechanisms using the Arima-HiC platform. Really, the function of this is just to sort of set the stage from a technology point of view to anticipate what we'll hear from the 3 fantastic speakers that we have lined up to follow. To get started, really I'm going to talk about HiC, Capture-Hic, and HiChiP, some of these different 3D genome techniques, either genome-wide or targeted. Not a lot about the performance of them per se, but sort of how they would be used and what the data looks like. And so you can think about how they might be useful for your research. But by way of introduction, just one slide here to introduce the 3D genome. I'm sure we'll hear much more about this in the talks to follow, but at a sort of a schematic level, if you will, the genome is organized into these hierarchical layers of genome organization. So if you can see my cursor up here on the left, we have chromosome territories that you would see if you look down at an interphase nucleus. Those chromosome territories depicted here in different colors is kind of what you would see in an interphase nucleus if you were to paint each chromosome a different color, and is also observed in HiC data, whereby the majority of the interactions that you capture from a technology like HiC are occur within a chromosome as opposed to between chromosomes. So you see this pattern across a HiC diagonal, and we'll talk more about how the data is visualized in a minute. And then as you kind of zoom in further here on the left, you'll see other structural features like parts of the chromosome associating with the nuclear lamina and other parts of the chromosome kind of extending more towards the nuclear interior, and other features like transcription factories. If you zoom in kind of another log order or so further, you see sort of on the megabase scale, roughly, these self-interacting chromatin domains that are seen here, which are demarcated by these architectural proteins that sit at the boundaries between these domains. So the concept here is if you sort of have 2 points, let's say A and B on a linear thread, if the points A and B were close together, but if the folding of the genome was, say, random, then A and B would also be close together in 3-dimensional space. But in many cases, that's not true, where the folding of the genome is not random, and A and B, even though they're linearly close along the genome, might be spatially segregated into neighboring gene regulatory architecture domains, like topological domains. And these types of self-interacting domains are often depicted as triangles, like so. Now, if you go back over to the left here and you zoom in, let's say another log order into the order of tens of kb or hundreds of kb, you'd see these sort of point-to-point chromatin loops that may be facilitated by architectural proteins like cohesin. And these types of structural events can also bring together cis regulatory elements like promoters and enhancers. Now, what does this look like again in Hi-C data? This is about a 7 megabase region that you see here. And these sort of triangles, if you will, of the Hi-C data would represent what one might call a topological domain. So you see two neighboring domains here. And then when you zoom in on one, these sort of puncta of frequent point-to-point interactions would be sort of consistent with this idea of a chromatin loop and these frequent interactions between the anchors of those loops. So why study 3D genome organization? Well, it's been studied now for decades, but in terms of technologies like chromosome conformation capture, more so over the past, approximately 15 years or so. And when it comes to Hi-C type technologies have been used in many different research areas, but specifically in the context of brain development, there's a couple studies here just to point out from Giacomo Cavalli's lab, as well as Dan Geschwind's lab, who we'll hear from later in the seminar, which was an analysis of human corticogenesis, which is an interesting study there. It's been analyzed really at a single cell level kind of recently by Joe Ecker and Jesse Dixon's lab at the Salk Institute in the human prefrontal cortex cell types. In neuropsychiatric disease from the Akbarian lab, which we'll hear from also during this seminar, as well as Yin Shen's lab at UCSF. And then also in the context of neurological genetic disorders like repeat expansion diseases, which has been studied in an exciting paper from the Phillips Kremens lab back in 2018. So to get down into sort of the Hi-C technology itself, some of the nuts and bolts questions that always come up are what are the supported sample types? And really we have a sort of a wide range of protocols. I think probably most relevant to this discussion will be human and mouse and, maybe flies or, other model organisms like that. We have user guides for cultured cells, primary cells, blood, fresh or frozen tissue. And in terms of the sample amounts, this is just a general recommendation, but, a typical Hi-C reaction will start with about 500,000 cells. If it's tissue, ballpark, we'll start with about 50 milligrams or so of frozen tissue. You grind that down and then you cross-link it. And then you'll probably use just a fraction of that for your actual Hi-C experiment. As well as 2 mils of mammalian blood, if that's the sample type. Now, the Arima-HiC workflow really contains of just a few sort of core steps that we've highly optimized. We start with cross-linked cells, as you see here. We're just trying to illustrate two pieces of DNA that are not really linearly close on the genome, this blue and gray piece, but they're being held together in 3D space by this protein. So if you kind of take that out and have a close look at just this one region, Hi-C consists of basically digesting the chromatin here with restriction enzymes. We use a cocktail of restriction enzymes that increase the cut site density across the genome. Those digested ends, as you can see here, that are produced are filled in with a biotinylated nucleotide and then ligated back together. And then you purify out that DNA. So the key part here is this ligation junction point where the blue and the gray have physically ligated together. That's the piece of DNA that really captures the 3D genome information that you're ultimately trying to detect. And then we finish with a quantitative and predictive experimental QC checkpoint that is predictive of the quality of the final sequencing data. And this whole process here happens in about 6 hours using the kit. So I've touched on the flexible sample input requirements. This whole process is incredibly simple and easy now. It's about 8 steps and the entire reaction happens in a single tube. You just keep piling on reagents. There's no spinning or washing or anything that would be particularly prone to sample loss. We have innovated, like I said, on the multiple restriction enzyme chemistry and the speed of the protocol compared to other Hi-C protocols that are out there. And then also the predictive QC checkpoints. Now, taking these molecules here that you produce from the kit, they have to be sheared and then prepared as a library for sequencing. And we haven't tried to reinvent the wheel on how to do library prep, but we've taken the approach of leveraging and validating a variety of different commercial library prep kits like the Kappa HyperPrep kit or the Swift kit or NEB, whichever you're comfortable with, you can use those kits and reagents and do a Arima-HiChiP library prep with them. Okay, so you enrich for these molecules here using biotin strictavidin conjugation. These are the ones that are going to be interesting for 3D genome analysis, the ones that are part gray and part blue. So you carry those into the library prep, you add your sequencing adapters, you amplify, and then you sequence in paired end mode such that read 1 will map to the gray piece of DNA in the reference genome, read 2 will map to the blue piece of DNA. And that's essentially one data point from one cell, from one interaction that ultimately goes in to feed the construction of the HiC map in a given sample. I should point out that the kit is quite compatible with low input cells. We say 50,000 cells or less. And I think the story that you'll hear more about later on from the Akbarian lab did conduct a Arima-HiChiP analysis on about 6,000 sorted dopaminergic nuclei, and perhaps we'll hear more about that later. And we do offer this technology as both a kit and also a service format. Just real quick to kind of benchmark the quality of the data, when we were first generating these Arima-HiChiP data, we really set out to try to benchmark it to the gold standard in situ HiChiP protocol that was developed by the Aiden lab, now at Baylor. So just one sort of example here, when we were looking at the published in situ HiChiP data in GM12878 cells, what we're looking at here is about a 1 megabase or so loop, right? This sharp, frequent interaction that kind of happens off the diagonal here. So this was generated from a really deeply sequenced data set, about 3.6 billion raw reads. And when we look at the Arima-HiChiP data set, this is only sequenced to about 1/3 of the depth, about 1.2 billion reads. But you can appreciate that the signal intensity at that looping region, in other words, sort of at the structural features that you're doing HiC in part to detect, is more enriched, right? So it sort of just speaks to the point, I know this is just one example, where the Arima-HiChiP data is able to detect loops at a greater signal strength, despite having here, less than half of the sequencing depth. And you can extend that analysis even further and say, well, what happens if we subsample this 1.2 billion reads down to 1 billion reads, or 800 million reads, or even down to 600 million reads, and then you call loops. And this line plot is simply just plotting the total number of loops that are detected in the Arima-HiChiP data at a genome-wide scale using open source tools. This is done with Juicer and HiCups, compared to the 3.6 billion read data set down here. And you can see even with a sixth of the total data, so 600 million reads in Arima compared to 3.6 billion in the published data set, you can still identify more loops. This isn't just internal data. Our customers have seen the exact same observation here. Okay, I won't go into Arima-HiChiP anymore, and just maybe point you to the testimonials page with others who have used the kit, even those who have had no HiChiP experience before or are working with really low precious input samples, perhaps derived from clinical specimens. Our website also has a swath of different publications in the epigenetics and genome architecture field that you can also browse. And where we're at as a company is we've been really, as sort of Joe said, we've been really happy and excited with the utility and the real benefit that several have experienced using the Arima-HiChiP kit. But at the same time, we also realize that genome-wide HiChiP is not the appropriate tool to answer every question related to nuclear architecture when it comes to NGS-based approaches. And certain hypotheses are better suited towards targeted approaches. And what we've been working on is developing those target approaches like HiChiP and Capture-Hic. So next I'll talk about Capture-Hic. I think kind of a nice way to frame Capture-Hic for those who might be unfamiliar with it is to position it relative to maybe a more familiar sequencing-based technique like exome sequencing, whereby in exome sequencing you have your cells, you extract the DNA, you usually shear that DNA down to a smaller size, you ligate your adapters, and then you amplify. And then instead of going into sequencing, you introduce these biotinylated oligonucleotides that hybridize to your desired sequences of interest. So in the case of an exome, these probes are complementary to the exonic sequences in the genome. They bind, and then you can enrich for those using biotin streptavidin conjugation, purify out the exome, amplify that, and then sequence it. And Capture-Hic is quite analogous to this, except the upfront DNA extraction part isn't just extracting genomic DNA, it's doing Hi-C and extracting the Hi-C DNA products using the Arima-HiChiP. So really you go through this entire Hi-C protocol that I described before: digestion, fill-in, ligation, DNA purification, and then the enrichment for the ligation junctions and amplification here. And then you add your probes just like you would for exome sequencing, right? But here, the molecules that you enrich, like let's say you were enriching for promoters, you would capture the promoter sequences, but you would also capture the distal interactions with those promoters, right, that were on the same molecule. Like if this was a promoter and this was some, distal region here. And then you amplify, and then you sequence in paradigm mode. And we've had a nice partnership with Agilent Technologies over the past year or so to integrate the Arima-Hic kit into their streamlined capture sequencing protocol and reagents. So just to kind of give a flavor for the different types of Capture-Hic that we kind of bucket these into, there's the concept of region Capture-Hic, where maybe you have a gene or genes of interest and you want to capture the topology right around those genes. You might design Capture-Hic probes right here to sort of tile the locus around a gene of interest and obtain that targeted Hi-C data right around those genes dispersed throughout the genome, right? The concept of variant Capture-Hic, where maybe there's a GWAS variant and the hypothesis is that it's perturbing some type of cis regulatory element, and you want to capture what that cis regulatory element is looping to as maybe a potential putative target gene involved in the disease pathogenesis. You might take an approach like this. And like I said, for promoter Capture-Hic, you design those oligos to the promoters, and then you capture those promoters, but also what those promoters are interacting with, both upstream and downstream. And the way that we work with customers on Capture-Hic is you can send us a set of genes and we can do the design to the promoters of those genes. You can send a set of GWAS SNPs, we can design probes around those, or just a set of coordinates like a bed file, and we can design probes around those as well. We will review that design with you until we're both happy with it, and then ultimately the probes will be ordered from another vendor, such as Agilent. Now, just to show, I think, one tidbit of data to show you what this sort of technology can be useful for is, we did an experiment here where we were interested in capturing the regions around a set of tumor suppressors and oncogenes that are listed here, like MYC, PTEN, and TREC. And the regions that I've listed here is essentially for like MYC, for example, we captured 3 megabases around the MYC gene. And for BCR, maybe 620 kb around the BCR gene. And in total, this is only about 0.4% of the human genome. So this design scheme would look like this. Here's BCR. Here are the capture probes. We enrich the region around BCR and we leave everything else behind. And we did this in 10 different sample types. Several of them are non-cancer cell lines, and then 7 different cancer cell lines of different tissue origins. So I think the real power to highlight here as just sort of one example of Capture-Hic is shown in this slide. And what we're going to be looking at here is in K562 cells, I'm going to show a Capture-Hic map around the BCR gene. And to make the point, I'm going to first show that data binned at low resolution. So this is about 25 kb bin size. And this is relatively low resolution, I'm saying, for what you can get with Capture-Hic types of approaches. And then here across the top is CTCF ChIP-seq data, K27 acetylation ChIP-seq data, RNA-seq data, and cohesin data, all sourced from the ENCODE consortium. And what you can kind of tell here is that, this is the linear genome being represented at this diagonal, and there's some type of interaction that might be occurring between this region on the genome with something down here. Like there's this increased red signal, which represents more reads between these two regions here. But when you trace that back up to what's going on along the linear genome, it's really sort of unclear where that anchor point of that interaction is. And importantly, the promoter of BCR has a CTCF and cohesin co-bound site, but there's also one actually just upstream of that promoter. You can't really resolve whether this interaction is associated with the promoter itself or not. You can take that same data and then bin the data at only 5 kb. So this is sort of like a higher resolution analysis of the same data. And again, now it becomes a bit more refined, and you can tell that there is some type of interaction occurring here. And when you go through the same exercise of tracing that up to where it is along the linear genome and what are the chromatin and transcriptome features of this region, you really still run into the same problem where you have 2 peaks of CTCF and cohesin, and you can't resolve whether this interaction is truly associated with the promoter or not. Now, you can cut this bin size again by a whole nother log order to about 500 base pairs, which is incredibly high resolution when it comes to Hi-C analysis. Now you can pinpoint, more precisely where that loop is. You trace it back to its anchor points along the linear genome, and now you can say with a lot more confidence that this loop is truly anchored at the promoter of the BCR gene. So this sort of, highlights, I think, one of the main advantages of Capture-Hic is if you just targeted this one region around BCR, which is about 620 kb, you could obtain this type of exquisitely high resolution Hi-C data for approximately 10 million sequencing read pairs per sample. So in conclusion, I hope I've been able to touch on the streamlined Capture-Hic workflow that uses Arima and Agilent reagents, the full support for the custom probe design, and then the ability to obtain the sub-kilobase resolution Hi-C maps for only about 10 million reads per sample. Now here in the last 5 minutes, I'll touch on HiChiP. And similar to Capture-Hic, I'll position HiChiP sort of relative to ChIP-seq, where I think most of us are probably familiar with ChIP-seq. You start with your fixed cells, and then you shear that. You add your antibody to bind to the protein target of interest. You do your IP, and then ligate on your adapter, amplify, and sequence. And HiChiP is just as it sounds in the name, Hi-C plus ChIP, where the first step is this upfront Hi-C reaction, using the same process of the restriction digestion, the fill-in, and the ligation, and then you shear, and then you add your antibody. So the molecule that's being immunoprecipitated here, the difference is it has the same sort of sequence content right around the protein target, but the molecule also contains that ligation junction that captures that 3D interaction that's associated with the target protein of interest. And then you just carry that through the sort of standard Hi-C library prep. So it captures this 1D protein localization information that you would get from ChIP-seq, but it also captures at the same time the 3-dimensional interactions that are associated with that immunoprecipitated protein. And some examples of that would be doing a HiChiP for H3K4 trimethylation to characterize the chromatin interactions associated with active promoters. And we do have optimized shearing protocols for the diagenode instruments as well as the Covaris. We had a nice collaboration with Covaris that was presented this past year at the AGBT conference, where you can look at that poster. And we also offer this technology as both a kit and the service format. Quickly, I don't have time to go through all of this, but HiChiP is a relatively nascent technology, but there's been some exciting applications in the field of neurobiology from Chris Glass's lab as well as Yin Shen's lab. And I just want to show maybe 2 quick snippets of data. This is the MIC locus here, and I've sourced a lot of ENCODE, ChIP-seq, and RNA-seq data around this locus. And the point that I want to make is, if you look down here at the bottom, this is the K27 acetylation ChIP-seq data, 2 different replicates of that, and here's the signal track. If you do HiChiP and you plot the data as sort of this 1D signal track, you still capture all of those same protein localization sites that you would get from ChIP-seq, and that's quite nice here. But what you're not getting from ChIP-seq is all the three-dimensional connectivity between those active K27 acetylation sites. So here is all the statistically significant loops that were associated with the K27 acetylation peaks across this locus. Here's the MYC gene here, and you can see a loop that extends over a megabase downstream to this K27 acetylation peak over here on the right. And qualitatively, these two replicates look quite reproducible. And when you quantify that at a genome-wide scale, about 85% of the loops that are found in Rep1 are also found in Rep2, and a similar number, about 80% on average, for K4 trimethylation HiChiP. Now we've completed beta testing to have customers validate the kit and the technology in their own hands. A couple of those key performance metrics for HiChiP is quite simple. Essentially, what fraction of your data represent long-range intrachromosomal interactions that could be useful for loop calling, and that's plotted on the y-axis. What fraction of the data are reads that overlap ChIP peaks as a metric of how well did the ChIP enrichment work. You want to be in this upper right quadrant here, and all 24 reactions from the beta testing program meet that criteria. And that high level of long-range interaction signal, as well as the high level of ChIP enrichment, helps you drive down the cost to obtain this type of targeted 3D genome information. So if you just count the number of reads that are required to obtain greater than 70% reproducible loops for the H3K4 trimethylation HiChiP, we're looking at about 100 million reads or so, and for the K27 acetylation HiChiP, about 300 million reads. And compared to, let's say, in situ Hi-C, where I believe the ENCODE recommendation is about 2 billion reads, the Arima-HiC, we recommend about 600 million reads, and then for HiChiP, you can see here, about 300 million for K27 acetyl, and about 100 million for K4 trimethylation. You can also look at our website from some of the testimonials from these early adopters of the technology. And in conclusion, I think we've developed a nice in situ HiChiP workflow that is quite robust at capturing 3D loops as well as the 1D chromatin occupancy data that you would get from ChIP-seq. About 80% of those loops are reproducible. It has been validated in customer hands across a variety of different cell types, as well as a couple different protein factors. And I didn't get a chance to talk about this much, but we do have a full end-to-end open source bioinformatics pipeline that we had worked on in collaboration with Ming Hu at the Cleveland Clinic, and a full range of technical onboarding for the bioinformatics so that you can analyze your own data with these open source tools. I'd like to thank our scientific advisors as well as the funding support that helped us develop the tools that you heard about today. And with that, maybe I can take one or two questions. Thank you.
Fantastic, Anthony. Thank you so much. If you could go back one slide, the very last point, sorry, one more. The end-to-end open source bioinfo, we received a question asking, does Arima provide Hi-C data analysis? Hi-C-seq data analysis?
As a service, do you think, or do you think the question was about just technical support?
I think it was about technical support.
Sure. Yeah, I can elaborate on that a little bit. Yep. So the typical process for someone who receives an Arima-HiC or Arima-HiChiP or Capture-Hic kit is step one is there's a technical onboarding call where our technical support scientists will get on a call with your experimental scientist and or bioinformatics scientist, and we go through all of the experimental protocols, and then we also go through installation, implementation, and interpretation of the open source bioinformatics tools. So for Hi-C, we support Hi-C Pro, Juicer, Hi-C Bench developed out of NYU. For Capture-Hic, we've been using Arima-HiChiP from the Babraham Institute, as well as Chicago and Schick Maxima for data analysis. And then for HiChiP, we've been advocating for a program called MAPS, which was developed by Ming Hu at the Cleveland Clinic, but we also support a program called FitHiChiP, developed by Ferhat A here in San Diego. So really a full range of support on installing, executing, and interpreting those data.
Yeah, fantastic. All complimentary.
Yep.
Next question. Maybe I missed it, but how much input material would you recommend for HiChiP in mouse cells?
Yeah, so the way that we think about input is it'll be approximately 2 to 4 million cells. Ultimately, what it comes down to is we try to input a number of cells such that the input is approximately 10 to 12 micrograms worth of chromatin. So that would be the target. You might be able to go lower depending on the protein factor, but a general recommendation is sort of in that 2 to 4 million cell range.
Okay, fantastic. Thanks, Anthony. And then I'll just answer, there's one more. Will the presentation be available for download to show my lab later? And the answer to that is yes. Our team will be in touch with the attendees, and we're happy to provide you that resource going forward. So thanks again, Anthony. That was fantastic. And moving right along, I'd like to introduce the next speaker in our series. We're very excited to introduce Dr. Schahram Akbarian. Dr. Akbarian is professor of psychiatry and professor of neuroscience at the Icahn School of Medicine at Mount Sinai. Dr. Akbarian's work explores epigenetic regulation of gene expression in order to identify epigenetic drug targets and novel treatment avenues for psychosis, depression, and other psychiatric disease. We're all very excited to have you, Dr. Akbarian. Thanks, and the floor is yours.
Hi, thanks the whole entire Arima team for giving me the opportunity to talk. And first of all, let me double check. Can you hear me and can you see my slide?
We hear you loud and clear. And I think if you put that slide presentation into presentation mode, we'll be good to go.
Okay, so now I press slideshow. Do you now see?
There you go.
Perfect, good. All right, good. And I guess the video camera doesn't work. I mean, I don't mind to show my face, but somehow I clicked on it and it doesn't seem to be activated, but that's fine. As long as you can hear me, everything is good.
We're good to go. Thank you.
Good. Okay, so I have about 20 slides, and I think the first 7 slides are science and the last 12 or 13 slides are technology based. So let me very quickly, as academic investigators, we're driven by the science. So I explained to you first what is the focus of our laboratory and how we use Hi-C in order to study the genomic architecture in the human brain. And we, as neuropsychiatrists, are limited to working with human postmortem brains, so it has its own limitations, and we often have grants that require sometimes hundreds of brains to be processed in fairly consistent manner. So some of the data you see today are probably not the most technically, not the latest hotshot technique, but it's more based on, the protocol working reliably so we can process many brains. So, so we started with some of our first Arima projects. We started with a medical problem that can be summarized as follows. So you have a lot of psychiatric diseases where the patients have, such as schizophrenia, for example, where the patient has a lot of medical comorbidity, like diabetes, hyperlipidemia, excess body mass, which is not a trivial problem. It contributes to the terrible 10 to 20 year reduction in life expectancy gaps for these patients. And this medical comorbidity is very hard to study on a genomic level because the genetic risk architecture of psychosis, including schizophrenia and metabolic syndrome, which summarizes a term of, diabetes, hyperlipidemia, and obesity-related syndrome complex, that there is very little overlap in terms of GWAS-based risk polymorphisms, et cetera, et cetera. So it's kind of, a typical situation where you have population level, very relevant medical comorbidity, but you have very little to study on the level of genetics. So we thought, to make a long story short, maybe we should test the hypothesis that lack of this shared GWAS-based genetic risk architecture does not necessarily preclude that there is no genomic interaction or shared chromosomal environment, including chromosomal conformations, in the nucleus of brain cells that are critically involved in the regulation of cognition and the regulation of metabolism. And there are some cell populations in the brain who have kind of double or triple duty, who are multifunctional. And one of them is, as many of you probably know, the midbrain population of dopaminergic neurons, which I try to express on this slide. So to the right, it shows, can you see my mouse? Do you guys see the mouse?
Yes, we can.
Okay, so you see this dopaminergic neurons project to the not only to the brain's reward circuitry, but also to the hypothalamic centers, which are critical for metabolism and nutrition. And on the left of this Zoom screen is the evidence summarized that probably already everybody knows about it as well, that links the dopaminergic neurons, both the neurons and its postsynaptic receptor systems, to the pharmacology of psychosis. So anyway, so we said, let's start out and let's examine this if we find something in terms of shared chromosomal environment of these, of these genetic risk architecture. So we had to develop a FACS sorting protocol for midbrain dopaminergic neurons, which works fairly well. To make a long story short, when you deal with postmortem brain, the nucleus as a physical entity is fairly well preserved, even 6, 20, sometimes 30 hours after death. It is a much more stable entity, so to speak, in the postmortem tissue as compared to synapses and neuronal processes, etc. So this nuclei can be even from frozen brain, because they hold together by their chromatin. They can be FACS sorted after immunotagging. We do this with dopaminergic neuron-specific transcription factors. Then we run a certain quality control like nuclear RNA-seq, and this shows some of these tracks. The red is from this FACS sorted midbrain dopaminergic neuron nuclei. Here's TH on the bottom right of this Zoom screen, right? Where my mouse is, tyrosine hydroxylase, clearly showing very nice enrichment for these Nur1 plus, Nurn plus. These are our dopamine neurons, and much less signal for some of these from these other cell populations. So we kind of developed this assay, and the only problem then for us was that we often got from a postmortem brain only 5 or 10,000 of these nuclei sorted. So it doesn't work very well with run-of-the-mill conventional high-C protocols, which, as you all know, require millions of nuclei as input. So this was a bit of a challenge, and we spent, I think, more than a year on figuring this out. And I will show you later in my slides the protocols that worked. And this slide just, the upper right is classical high-C interaction matrix, TAT, topological associated domain landscape, is really only there to convince you from the data that the protocol works, that we are able to get nice high-C data out of these very limited numbers, 5 to 10 or maximally 5 to 50,000 nuclei at best, input material from this postmortem brain. So we're very delighted. And then the rest is fairly straightforward. We looked for topological associated domains, chromatin domains, that even if they don't have overlapping genetic risk sequences, risk polymorphisms for excess body mass index or BMI and schizophrenia, we looked for topological domains that have somewhere in the domain risk polymorphisms for one conditions, and another portion of that same domain risk polymorphisms for, in this case, schizophrenia. So, and by doing that, we then put the whole high-C data set into this Chrome 3D genome modeling algorithms that has been developed a couple of years ago, I think by a group in Scandinavia, and it works fairly well. And what you see here on the upper, and this was work done by Sergio Espeso-Gil, what you see here on the upper left, this multicolor, is each color is a chromosome. So this is basically the Chrome 3D solution after 1 million or so Monte Carlo based iterations of this solution of the of Chrome 3D, how these topological asset domains are arranged. Each of these beads, each of these little beads is one domain, and this resolution is still not ideal. It's not the same fine resolution as with if you have an input material of 1 or 2 million nuclei, but it's good enough. It was good enough for this purpose. And then what the bottom left shows in red are each of these red beads is a chromatin domain that harbors both risk polymorphisms for schizophrenia and risk polymorphisms for excess body mass in that same domain. And then what Sergio did, he did some Euclidean distancing measurements and found out, much to his delight, that there are at least 10 locations inside these nuclei of these dopaminergic neurons where chromatin domains which share these risk variants for these both conditions seem to form kind of what he calls Euclidean hotspots, basically a non-random clustering of multiple domains, sometimes from different chromosomes, that seem to basically coalesce in a non-random manner. And when he looked for the type of genes that are expressed in these domains, this is in the upper right of these Zoom slides, there is a number of very interesting signaling pathways, brain-derived neurotrophic factor, BNF, and kinase signaling pathways. When he looked for, this is on the bottom right of this slide, when he looked for transcription factor motifs, there was enrichment for dopaminergic neurogenesis, white fat cell differentiation, and so on. So this was all quite exciting, and for us, one of the most striking examples of the promise of this technique were certain Euclidean hotspots, where I'm jumping in the interest of time because I want to talk about the technique. One of these neighborhoods of risk-associated chromatin domains included sequences on chromosome 16, chromosome 17, and 22. And what's the cool thing here is that it captured a well-known locus on chromosome 16p, which harbors copy number variants, where the patients are in some ways psychotic or otherwise cognitively impaired, and they're all either abnormally, they have either basically abnormally thin or abnormally sick. So they have always disturbances in body mass, and it can go in both directions. So it's a very interesting and mysterious locus on chromosome 16p. And much to our delight, our Hi-C assay identified that this locus is heavily interconnected, or has a lot of, I should say, spatial proximity in this nuclei of these dopamine neurons to loci on chromosome 17 and 22, where master regulators, master transcription factors for cholesterol biosynthesis and triglyceride biosynthesis are located. And these transcription factors are well known in the metabolic field under SREBF1 and SREBF2. So this is the kind of example, I think, or promise of using Hi-C data to map spatial topography of chromatin domains inside the nucleus and develop new hypotheses along the lines I just mentioned. So I don't want to go too much into more of this science, and I want to emphasize that by now we have, I think, a fairly good handle on how to do Hi-C in these postmortem tissues within this context I just explained. And we basically have two favorite protocols for two very different applications. We developed one protocol, both of these protocols are based with Arima-HiChiP kits by now because they're very convenient for us to use. And I'm saying this, I'm not paid by Arima or anything, so right, I don't have a conflict of interest. It's just happened that they're for us quite straightforward to use in the lab. And so the top protocol is based on this, this basically what I just have shown you as in this first part of the talk. This is this more challenging protocol where we FACS sort nuclei from very small brain regions, such as the ventral midbrain. Hang on. And so this only involves a small number of nuclei, 5 to 50,000 nuclei at best, limited input material. The other protocol is more standard, where we take larger chunks of brain tissue, such as, for example, from the prefrontal cortex, and then use this material, which basically includes 1 or 3 up to 3 million of nuclei, to do the standard Hi-C protocol. So all these protocols involve, at the beginning, have a very similar path, cross-linking, lysis. In case of these dopaminergic neurons, we have to do a FAC sorting in between, but the big differences come in the next few steps. So we have modified a little bit of what has been described as to do from what has been described as to do in the Arima-HiC kits, and Marina and others from our team are very happy to provide you with some more technical details. I'm skipping a little bit over this slide because I think the key differences are here, and we come back to that later. So in what we basically do is we increase, when we generate the libraries from these kits, we change a little bit the cycle number. So we increase the PCR, number of PCR cycles when we prepare the libraries to 14 cycles for the low input protocol, and keep it to 10 cycles for the larger numbers of nuclei. And what it boils down to is, much to our delight, when we send these for sequencing, first by MiSeq, and then if the MiSeq looks good, we sequence deeper with the Illumina NovaSeq system in these days. So the cis-trans ratio is, even for the low input protocol with a small number of FAC-sorted nuclei, still pretty good, actually. I have to say it's up to 50 or sometimes 75% of all chimeric products are in cis, so from the same chromosome. This is a very, very rough quality index. So the cis-trans ratio is pretty good, as expected, 2 or 3 to 1, both for the midbrain and for the homogeneous samples, and same thing for the overall number of chimeric reads. The big difference is in terms of things that you normally don't want to see, like PCR duplicates, dangling ends, etc., etc. So when you look here on the right of this, this is a big difference, right? On the right of this slide for the prefrontal tissue homogenate, you see that most of these sequences that come back from the sequencer are of really high quality, and there's a very little PCR duplicates, etc., which basically this is what you would expect from a standard Hi-C protocol. While if you use our low input Hi-C protocol on the left, you see that the number of duplicates and other artifacts is definitely higher than for the conventional protocol, but it's still, for us, good enough to generate high quality Hi-C maps in general from these, even with this material. And so this shows just some readout in terms of cell type specific separation between dopaminergic and non-dopaminergic neurons. I think I can go over this because in the interest of time, it's now already 20 minutes. And, so this is probably the last slide, just to summarize the principal difference in working with homogeneous and millions of nuclei versus limited samples with just a very much smaller number of nuclei. But both protocols work, and we can basically recommend and happily to share both protocols with the wider field. And with that, I say thank you for listening.
All right, thank you very much. One question here for you is, I think, regarding, I think generally we hear less about interchromosomal types of interactions, but you showed that example on your slide of those interchromosomal links between the domains that have both metabolic and schizophrenia risk variants. What do you think would be some of the underlying mechanism of that co-association in the nucleus?
Yeah, that's a good question. I think in general, I think there are two answers. One is, if we take a regular Hi-C data set and we ask for transchromosomal contacts, the only way that we can define them reliably is if we have very large window sizes. So if you have, for example, 1 megabase wide bins, you can, even at very high confidence, call transchromosomal contacts, but that's not of interest for the majority of questions, right? So what happens is, what we did in our case, we first did, as I said, this Chrome 3D genome modeling that uses Monte Carlo iteration and some basic parameters from chromosome polymer physics, I believe, to come up with a solution of having the closest distance without breaking the chain, you know, of this TAT interactions. And once you have the Chrome 3D model for the internuclear topography of all of the TATs in the genome, and once you have that, you define these Euclidean hotspots and say, this is now a group of TATs that are closer together than you would expect from a random model. And then within that group of TATs, you look for these transchromosomal contacts. So it is not anymore against the genome-wide background. And that would be the best answer I can offer. I should also say that this is, in my opinion, only a first step of further, you know, developing the model by doing, for example, DNA FISH and other experiments to validate the spatial proximity of these TATs and any locus to locus transchromosomal contacts within this cluster of TATs from these different chromosomes.
Great, thank you very much. One other question here from from Dan from the virtual audience is, could you please comment more on the TN5-HiC technique that you mentioned on one of your slides, please?
Yeah, so that's a good catch. And I, again, in the interest of time, I didn't want to talk about it. So TN5-HiC was initially a protocol that we developed in-house to accomplish this challenge of generating HiC maps from small numbers, a few thousands of human brain extracted nuclei. It basically was a protocol that is entirely based on restriction, digestion, ligation, and transposase-based fractionation of the chromatin without biotinylation. And it was a protocol that gave us, I mean, enough material that we published this genome medicine paper and some of these TAT maps, Chrome 3D maps I showed you. We by now have retired our in-house protocol, because this modification of this commercial kit, of the Arima-HiC kit, gives us a much better, in general, better cis-trans ratios, and in general, higher percentage of artifact-free chimeric reads than our in-house protocol. And so that's, if you're interested in the transposase protocol, I mean, we can send it to you, and it's all documented in the genome medicine paper. At this point, I wouldn't recommend it as a first choice because it just has a poorer signal to noise in general, in part because of the lack of the biotinylation enrichment.
Excellent. Dan says, thank you very much. I think now would be a good time to, maybe Joe, would you want to introduce the next speaker, and we can all thank Dr. Akbarian for an excellent presentation.
Yes, so much. Thank you, Dr. Akbarian, and thank you, Marina, as well. Fantastic. So moving right along, I'd like to welcome our next speaker, Dr. Jon Beagan. Jon is a member of Jennifer Phillips Kremen's lab at the University of Pennsylvania. The Kremen's lab investigates the epigenetic mechanisms regulating development and function of the mammalian central nervous system to discover how genome architecture controls genome function. We're extremely pleased to have you here today, Jon. Welcome.
Thanks very much. Please let me know if you can hear me and see my slides okay. And yeah, so I'm Jon Beagan. I'm a recent graduate of the Kremen's lab at UPenn, and my PI, Jennifer, couldn't be here today, but I'm thrilled to take her place and really honored to be amongst such distinguished speakers. So thank you again. And yeah, I'm thrilled to present today a story we've been working on for the past few years, and it is set to actually come out public next week. So be on the lookout for that. And really, generally speaking, our lab is captivated by the complexity of neural connections, how they are specifically wired, and then how the connections are maintained, strengthened, and plastic throughout the lifetime of an individual. And the overarching question that drives our research is how genetic mutations and epigenetic modifications due to the environment will govern synaptic plasticity in both healthy mammalian brain activities as well as neurological disorders. And of course, I wouldn't be speaking today if we didn't particularly focus on the regulatory role 3D genome folding plays during these processes. And so in particular today, I'll be focusing on one feature of neurodevelopmental biology, and that's how neurons alter their behavior and functional output in response to input signals, i.e. how neurons rewire during development. So here we're looking at the visual cortex of a mouse responding to different visual cues, and each flash of light is a neuron firing an electrical impulse. And on the surface, this is amazing, but why is this phenomenon interesting as it relates to chromatin folding? And that's because as we were digging through some seminal literature, we kept coming across observations like this one. So here we're looking at electron microscopy images that show upon neuronal depolarization or neuronal activation that global chromatin folding undergoes marked changes in compaction that get stronger during depolarization or as depolarization is sustained, and then can recover upon removal of the stimulus. But of course, this kind of approach doesn't give us genomic location information. So we wanted to understand how genome topology connecting specific distal regulatory elements is reconfigured during neuronal activation, and that would be using C technologies like what have been discussed by the previous speakers. And we believe that this approach, this goal of identifying genomic locations, specific genomic locations that rearrange during neuronal activation may be important because of the potential that this may be a regulatory force overseeing activity response gene expression in neurons. And so by this, I mean, when a neuron undergoes one of those depolarization events in response to an external stimulus, calcium ions flow into the cell body through voltage-gated channels that triggers a signaling cascade that makes its way to the nucleus. And this leads to the upregulation of a set of genes I'll be referring to as immediate early genes, or IEGs, such as FOS and ARC. And this really occurs on the time scale of seconds to minutes. It's incredibly rapid. And some of these genes, like ARC, go on to encode proteins that can then go and alter the synaptic properties of the neuron, whereas others, like FOS, encode transcription factors that drive the upregulation of a second set of genes, which I'll refer to as secondary response genes, or SRGs, such as BDNF. And this upregulation of BDNF, or these secondary response genes, occurs really on the time scale of minutes to hours. It's much slower in comparison to IEG expression. And then, of course, these genes like BDNF encode proteins that can go back to alter the functional properties of these neuronal networks.
Hey, John, sorry to interrupt real quick. This is Joe.
Sure.
It's a little bit choppy. I don't know if you can do anything to affect the connection. It's still, okay. It's still comprehensible, but just a little choppy, just FYI.
Okay, I'll just slow down a bit, and hopefully it catches up. Sorry about that.
No problem.
So this activity-dependent gene expression regulates how synapses form and degrade and function, which is especially critical during those early developmental years as our brains are shaped by our environmental stimuli. But activity-dependent gene expression continues to be critical for how we store memories and process thoughts well into adulthood. And so my PI Jennifer and me were really fascinated by all of this and anchored on three basic questions at the start of our work. First, are genome folding patterns altered upon the stimulation of neural activity when we use C-based approaches like Hi-C compared to those electron microscopy images we were looking at earlier? Second, are there different long-range 3D epigenetic mechanisms that regulate IEGs, those rapid response genes, compared to those much slowly responsed SRGs? And finally, what is the timing of loop formation if there are any dynamic loops? And how does that relate to the functionality of those loops? i.e., how does that relate to the expression of the genes at the base of those loops? And so we set out to answer these questions using chromosome conformation capture carbon copy, or 5C. And much like Capture-C, that selects specific regions of the genome across which we can analyze global chromatin folding patterns. And so we created these high-resolution maps of genome folding in murine cortical neurons after inhibition and induction of neuronal activity. And again, this is just across a select subset of the genome. So we did this surrounding key immediate early genes and key secondary response genes that we were interested in, as well as other synaptic proteins that were not as dynamically expressed across neural activity. And so our first pass impression from these maps is that genome folding at a global scale is largely unchanged upon alterations in neural activity. So here we're talking about those higher levels of the genome folding hierarchy. At the TAD level, at the compartment level, we see very little change across these maps. And it's important at this point to highlight that this is kind of what we expected to find, to be honest, that most of the previous literature on dynamic genome architecture, including our own work, was focused on very large scale changes in cell state taking place across several days. And so here, we're just looking at changes in neuronal activity of post-mitotic neurons on the time scale of minutes to hours. And so we knew, we kind of came in expecting not to see these huge scale changes, and we knew we wanted to focus on specific punctate looping interactions. But it was possible and even probable to us that loops were unimportant here, or at least not dynamic. And so we came up with 3 possible models for what we would find that we could then test. So first, the first hypothesis, this nearest enhancer hypothesis, is that loops really were perhaps unimportant here, that only activity-induced enhancers, this blue orb here, that were close enough to genes such that they did not require long-range looping interactions, that only these enhancers actually drove activity response gene expression. Second, and kind of my personal favorite hypothesis, was that perhaps loops were important, but invariant. So the same loops existed in inactive neurons as they did in active neurons, and they actually poise these activity-induced enhancers near their target genes so that when the neuron becomes active, these enhancers can turn on and rapidly upregulate the target gene without the need to form a new loop. And finally, we were open to the hypothesis that both enhancers and loops would form together as neurons become active. And so first, we wanted to test the validity of this nearest enhancer model. And we did that by building 3 simple linear regression models. And so in each of these that I'll show, we're using the H3K27 acetyl CHIP fold change signal at different regulatory regions as the explanatory variable in these linear regression models to predict the response variable of gene expression. So here, this first model, this positive control, as it were, I'm showing using promoter acetylation fold change as the predictor of gene expression fold change. And like I say, this is kind of our positive control because we really expected and indeed saw a strong positive correlation between the two. And this simple model explained roughly 50% of the variance in our transcription data on its own. And then we tested the relative contribution of different enhancer elements by adding them as a second explanatory variable term in these linear regression models. So here, on top of this promoter term, we're adding a second term that contains the acetylation fold change of just the enhancer that's nearest to each gene along the linear genome. And actually what we saw is when we add this term, it does not show a strong positive correlation with gene expression, and it does not explain much more of the transcriptional variance in our data. And this is in contrast to when instead of the nearest enhancer, we add in the term of the looped enhancer acetylation fold change for each gene. This does show a much stronger positive correlation with gene expression and explains much more of the transcriptional variance that's held within our data. So we use this data to suggest to us that loops do indeed matter for regulating activity responsive gene expression. And at that point, we focused on distinguishing between these 2 looping hypotheses. And really, to do that, it was pretty simple. It just involved looking for the presence of these dynamic loops within our 5C regions, within our 5C analysis data. And we did this using a computational package developed in the lab by two fellow grad students, Thomas Gilgas and Lindsay Fernandez, that identifies dynamic looping events in an FDR-controlled manner by changes in loop strength across conditions. And actually what we found was that a small but notable proportion of the loops we identified, actually about 10% of those identified, did not exist in the repressed activity state and were de novo induced upon neuronal activation. And oftentimes these dynamic loops, these activity-induced loops, connected those activity-induced enhancers via these loops to specific target genes that were upregulated, oftentimes IEGs. And one example of that, again, is this FOS gene that I mentioned previously, where specifically in active neurons, the FOS gene on the x-axis here loops upstream to this activity-induced enhancer region. And we believe drives the upregulation of the FOS gene. And so in a more quantitative approach, rather than just looking at specific examples, we can lump all of those genes together that form this set of dynamic loops and look at their expression profiles. And again, I'll remind you, this is just a subset of the genome, but even within our selected subset, these dynamic loops were relatively rare, but the genes at their base were all highly upregulated in active neurons compared to inactive neurons. And so we classify these loops as rare, but with a strong correlation towards transcriptional upregulation in active neurons. And again, this is in contrast to those poised loops, those invariant loops across activity states that connect activity-induced enhancers to target genes, which were much more common, but the genes at the base of these loops were only modestly upregulated in active neurons compared to inactive neurons. And finally, as sort of a negative control, we included those loops where at their base are enhancers whose activity actually goes in the opposite direction. Their enhancer activity goes down in active neurons. And as you might expect, the genes at the base of these loops, their expression also goes down in active neurons. And so we concluded from this data that both dynamic and poised loops regulate the activity response gene expression, although in different abundancies and degrees. And at this point, we return to our question of whether different long-range epigenetic mechanisms regulated those rapid expressed IEGs compared to the slowly expressed SRGs. And we had already seen that these dynamic loops, this first class of loops here, connected the IEG FOS to a nearby activity-induced enhancer. So we asked whether these similar dynamic loops regulate the expression of an SRG like BDNF. And indeed, we do observe similar dynamic loops formed by the BDNF gene. So here's BDNF on the x-axis. And it's looping upstream, again, specifically in activity-induced neurons to this set of activity-induced enhancers. But visually, it was instantly clear that BDNF actually had a very different overall looping structure than FOS. And you can see here on the left, this specific loop is actually a zoom in of this very complex looping structure that BDNF forms across this roughly 3 megabase topologically associating domain. And we were really blown away by how complex this looping structure is, especially in comparison to that relatively simplistic looping structure that FOS exhibited. And so ultimately, the lesson here that we learned from this initial 5C data set was that the immediate early gene FOS engaged in very simple singular loops to a very small number of enhancer targets, whereas BDNF engaged in a complex network of very long range loops connecting to many different distal activity dependent enhancers. And given that we know genes like FOS are activated rapidly, whereas secondary response genes such as BDNF are expressed much later, our leading hypothesis is that looping complexity is linked to timing. So we hypothesize that these immediate early genes in general, like FOS and ARC, form simple short-range singular loops, and this helps the gene get activated quickly, whereas secondary response genes form multiple intertangled, much longer range network of loops, connecting to many enhancers, and this is connected to their delayed expression profile. And although I don't have time to discuss all of the details, I will direct you to our upcoming manuscript, where we do find that these trends seem to hold true across genome-wide IEGs and SRGs in general. That SRGs form more loops, longer loops and interact with more enhancers. And at this point, I'll just point out, because we're in the process of profiling these IEG loops genome-wide, because they're very short range, we need that high resolution genome folding mapping technology, like the Arima-HiChiP maps that we've seen in the previous presentations, to identify such short range dynamic looping interactions. But this work did not answer one of our initial questions as to whether these longer SRG loops also display delayed formation kinetics. To answer this question, we mapped genome folding, enhancer activity, and gene expression across an acute timescale of neuronal activation. So here I'm showing again the FOS gene, the same heat map I showed before, gene expression of the FOS gene, and then also enhancer activity of these two different activity-induced enhancers. And here we're looking at 0 minutes of neuronal activity or inactive neurons. And again, we see the gene is off, the enhancer is off, and no loop has formed. This remains true after 5 minutes of neuronal activity, but by just 20 minutes of induced neuronal activity, and really this kind of blew our minds, we see this loop form very strongly. This enhancer is on, it's very active, and the gene has begun to be turned on. But it's not until 60 minutes of neuronal activation that we see the gene is fully expressed, and the loop remains strong, and again, the enhancer remains active. And finally, by 6 hours of sustained neuronal activation, the gene has begun to be turned off, the enhancer is inactive, and the loop, some signature remains, but is basically disconnected. So this was really striking to us that this loop could form so rapidly, and in fact, the loop formation precedes peak mRNA levels. And as we're beginning to dissect or do causal experiments, we're still dissecting the regulatory role of these loops, but this to us was an initial indication that this loop is in fact driving the expression of the FOS gene. And we can compare this FOS dynamic looping to the looping kinetics at BDNF, that SRG that I've been discussing. So at BDNF, like FOS, there's no looping structure at this dynamic looping heat map in inactive neurons or at 0 minutes of neuronal activity. And this remains true like at FOS at the 5-minute time point. But really, this remains true through the first hour of neuronal activity, and we don't start to see looping signature at this site until 6 hours of sustained activation, at which point the gene has also begun to be turned on. And we don't see maximum loop strength and maximum gene expression until that 24-hour time point, full day of sustained neuronal activation. And so obviously this is different than that FOS locus that I showed previously, where this is much slower loop formation kinetics that's much more tightly correlated with the gene expression timing, and loop formation does not actually precede the maximum mRNA levels of this gene here. And so in summary, what I've shown you today is that greater than 10% of the loops we identify within our 5C regions actually are induced de novo upon the activation of cortical neurons. That these activity inducible enhancers engage in those poise loops and also those dynamic or de novo loops that connect these enhancers to genes that respectively are modestly and very dynamically upregulated in response to neuronal activity. Also, that immediate early genes like FOS and ARC connect to target enhancers via very short-range, simple looping structures, compared to secondary response genes like BDNF, which engage in a much more complex network of long-range activity invariant and activity-inducible loops with much slower formation kinetics. And just before I wrap up, I'd just like to introduce some of the future directions we're taking this work. As I mentioned, we're now beginning causal experiments to dissect the regulatory role of these enhancers and loops. And so that involves both using CRISPR-based technologies to disrupt these enhancers and loops, as well as using loop engineering techniques like Ladle, which was developed in our lab recently, to induce formation of these loops in activity states when perhaps these loops or enhancers do not usually exist or are not usually active, to map their regulatory potential in expression of these target genes like FOS. And finally, we're beginning to use our activity dynamic enhancer sets to map their preferential locations at loop anchors with neuropsychiatric disease-associated genetic variants, thereby mapping target genes that may exhibit activity dynamic expression patterns and make predictions about the activity states in which genetic variants may disrupt expression. And with that, I just, again, want to acknowledge my amazing mentor, Jen, who wishes she could have been here to give this talk herself, and our awesome collaborators at the University of Utah, Dr. Shepard and his postdoc, Dr. Pastusan, my co-authors here highlighted in purple, and all of the members of the Kremens Lab team, as well as our funding agencies. And with that, I'd be happy to take any questions.
All right, thank you very much, John. We have a question here from Gianni. I think I have this question as well. And that question is, for a neural activity response gene, if you have multiple loops, then how do you decide which loop to use for your gene expression association analysis?
Yeah, great question. So we thought a lot about that and actually tried a few different approaches. And the one that worked best was actually a slight adaptation of the ABC model that was recently published out of Jessane Greitz's group in Nature Genetics. And so that's basically multiplying enhancer activity by looping strength to get a prediction of the enhancers that matter most to regulate each gene. And so we made slight adaptations to that. We used thresholded loops rather than just high C contact frequency in general. But for each gene, it was always the enhancer with that highest loop strength times enhancer activity score that we paired a given enhancer to a specific gene.
Got it. Okay. Yep. That makes sense. I was also wondering, you know, I think one of the things that you wonder when you have a locus like this for, let's say, the SRG, the secondary response genes, when you see contacts between a promoter and multiple enhancers, you know, I think the word network sometimes implies that it's a co-association or a hub of interactions, but do you have any idea of whether those are just pairwise interactions and heterogeneity across cells, or are they forming actual hubs of one promoter and multiple enhancers?
Yeah, that's a great question, and you make a great point that so I didn't stress this really, but at this point, these are just whole cortices that have been dissociated and plated in vitro, and then their activity manipulated with drugs. And so we do have a heterogeneous cell population. And so we do believe some of these loops and enhancers may be cell type specific, and we're picking up an ensemble of loop signature across the cell population. And so many of them may be, you know, present in this subpopulation, but not another. So many of them may be cell type specific. I don't have a good answer for you on in terms of if multiple loops are present in a given cell, are they hubs? We haven't tested that, and I think that's a really interesting question. And one future direction that we've discussed getting around these questions is using like an oligostorm or like an ORCA approach to actually image some of these TADs and loops within single neurons within a neural population as we manipulate their activity. And if we could correlate, you know, single cell folding imaging in that way with activation of specific neurons, we could maybe begin to answer some of these questions.
Okay, thank you. Let's see if we have any. Okay, one other question here is, I think you mentioned something, John, about broadening the analysis of these early genes and secondary genes to a genome-wide scale. Could you comment more on that and any findings from that genome-wide survey and how it relates to what you presented here?
Yeah, so we haven't necessarily done genome-wide analyses across activity states, but we could look at genome-wide looping signatures of just actually inactive neurons. And so we're more looking at those poise loops, those invariant loops, and the formation of those loops by those different gene classes. And so using that inactive neuron Hi-C data set, we could then classify or quantify how many loops each SRG formed, how many loops each IEG formed, and then compare the two. And so, like I mentioned in my talk, when we do that global analysis across the genome, we do overall see that SRGs in general form more loops, form longer loops, and interact with more enhancers than IEGs. And I will say we were leveraging, to classify IEGs versus SRGs, we're leveraging a data set from the lab of Jesse Gray, and they actually take it a step further to classify SRGs or delayed response genes in their case based on translational dependence versus independence, or the dependence of new, translation of new proteins to drive the expression of these delayed response genes. And so what we actually see is that those genes that are not dependent on novel protein translation to be upregulated in response to neuronal activity, that those are the genes with this really dramatically increased looping signature and longer loops, more loops, that more loop complexity signature that I mentioned. And so we really do believe it's a specific epigenetic state linked to this class of genes that display this delayed expression profile in response to neuronal activity.
All right, one more technical question here. I guess I was also wondering a similar thing is, I think for some of these looping analyses with the 5C data, and maybe this also translates to Hi-C data, is what type of resolution or bin size do you think you have to analyze the data in to observe what you did here in your own study?
Yeah, this was actually our first study in the lab where we used an updated 5C approach, which called the double alternating 5C approach, which has 5C primers designed to both ends of each restriction fragment. But we still use Hindi3 as our restriction enzyme, so we're kind of limited by how big those restriction fragments are. So we tend to bin our data at 4 to 5 kb, depending on the regions we're looking at. And because just of the size of the restriction fragments that we're working with, that's kind of the limit of our resolution. But if you go to say, a 4-cutter or a really high resolution cocktail of restriction enzymes like the Arima cocktail, you can obviously improve that resolution. You just have to, in the 5C approach, design more primers because you have more restriction fragments and you're designing primers to each fragment. And so that drives the cost, your initial cost up. So, it's a give and take there. Hopefully that answered your question.
Yeah, that does. Okay, well, thank you very much, Jon.
Great, I'll stop.
Really great presentation, great discussion on the questions, and I'll hand it back over to Joe to introduce the next speaker.
Great, thanks very much.
Yeah, thank you so much, Jon. That was fantastic. And thanks to the 3 previous wonderful talks, and we have one big one to go. Finally, we're thrilled to welcome Dr. Daniel Geschwind. Dr. Geschwind is the Gordon and Virginia Distinguished Professor of Neurology, Psychiatry, and Human Genetics. He's a Senior Associate Dean and Associate Vice Chancellor for Precision Health at UCLA. Pioneering work in the Geschwind Lab integrates population genetics, functional genomics, and bioinformatics with basic and clinical neuroscience to advance our understanding of neurologic and psychiatric disease, pathogenesis, and to accelerate treatment development. Dr. Geschwind, we're incredibly honored you're here, and the floor is yours.
Hi, thanks. I don't know if you can hear me.
Yes, sir. We got you.
I'll try to share the screen then. We'll see what happens. Let me find my There we are. See if this works. Can you see anything?
Yeah, we see your entire desktop. I think when you
Okay.
share screen, you just need to select the
No,
presentation.
that's what I've done. I thought that's what I'm almost positive I did. I just selected this Arima. I see. That's weird. See if that works. Is that better?
Here we go. Yep. Now just presentation mode and we're good to go.
Okay. It's in presentation mode, but you might be seeing the wrong display. Just to tell me if you are. What's going on here? That's weird. I started it. Oh, it's just my PowerPoint is sleeping. Do you see the shared screen, or what do you see?
We see the first slide and then the second slide.
Okay, good. I'll just swap displays. How's that?
That's perfect.
Okay, so I'm going to talk about just to give a little background. It will be obvious. My lab is not an expert in chromatin biology at all. We're interested in understanding how human genetic variation actually impacts the brain and brain diseases. So we're trying to use these approaches to connect genetic variation that increases susceptibility for human brain diseases, mostly neuropsychiatric, to understand their genetic mechanisms, and then of course eventually the kind of cellular molecular mechanisms. So I'm going to give a kind of high, at some level, maybe I'll go into some details in some areas, but mostly go over what we've been doing. And I wanted to acknowledge key people in my lab. This is a newer picture on the right that doesn't include an essential person, Hae-Jung Won, who really started doing Hi-C in my lab and brought Jason Stein and Luis de la Torre Viedo, who worked with her initially and also both in Hi-C a little bit, but more so in other chromatin explorations. It will become clear using ATAC-seq and other data. All the starred people now are gone. Hae-Jung and Jason are both at UNC Chapel Hill in genetics and neuroscience with their own separate labs. Luis is at UCLA. Neil's in a neurology residency. Mike's at UCLA. Vivek's at UCI. And then I'm going to talk about some work that's unpublished that Jenny Yin's been working on, who's a very talented postdoc in the lab. And some of that work has been done in collaboration with Alison Goat at Mount Sinai School of Medicine. So I wanted to put this first in case I run out of time, because I tend to. And of course, we're grateful to our funding sources, and we've been, a lot of what we're doing now is incorporated within the PsycEncode project. My talk's going to be in two pieces. One, I'm going to talk about neuropsychiatric and developmental disorders, and the second, exploring the impact of structural variation. So just to start with the first part, neuropsychiatric diseases have strong genetic components. Their prevalence is high. Their twin kind of family-based heritability and SNP-based heritability is significant and substantial. And the main point I want to make here is that the genetic contributions are complex, and that as the sample sizes have increased, shown on the left in the y-axis in D, schizophrenia being the big one, there one can see it's not quite a monotonic relationship at all, but there's a relationship between the number of samples studied and the number of loci identified. So loci. So anyway, the point being that there's a lot of gene finding going on. It's really highlighting the complexity of these disorders. And this is just showing some work in autism. That's just a summary from a few years ago that shows not only are we identifying common variants through GWAS, but especially in conditions like autism, the earlier, more severe conditions, we're identifying maybe up to 20% of patients who have a large contributory, some people might say causal mutation. And these are comprised of large effect size mutations in a number of genes, none of which really account for more than 1% of the syndrome. You can see in parentheses what was the contribution at the time. So it's a kind of this 20% is even a collection of rare disorders, and we can even put on top of that common variation. So we have a complex picture, but our job is to kind of embrace this complexity because it is what it is, and then to understand mechanisms of disease from this. And I think we have all the tools in hand to begin to do this. So I'm very optimistic. We just have to not put our head in the sand. So advances in genetics and genomics now make it possible to identify genes involved in susceptibility for common neuropsychiatric disorders. The same goes for neurodegenerative disorders, et cetera. However, they highlight extreme heterogeneity, even in kind of rare, conditions with rare forms like autism. Individual genes don't act alone. They act within pathways and networks, and they're obviously genetic sequence effects are modified by these other factors. And so our approach the whole way along has been to scratch our head and try to use integrative systems biology approaches to identify pathways and networks and translate genetic findings into mechanisms. And, and we have all kinds of interesting questions around this. Will we, despite the heterogeneity at the etiologic side, are there areas of convergence like there are in most disorders? And the answer is likely yes, but we don't know exactly where and when this will occur yet. But this is one of the main things we've been working on. So I'm going to start with just showing you some of that so you can see how that informs what we're doing with epigenetics. So this first paper came out in 2013, and it was a study that was based on scant genetic information at the time. In 2012, a series of exome sequencing papers came out in autism that I think combined had fewer than 1,000 probands. And we took those genes, which were, that were hit at least once with protein disrupting de novo mutations, and asked if we created developmental brain networks and normal brain development and looked at the trajectory of the transcriptome, could we identify, were these genes randomly distributed or were they distributed in very specific pathways? And it turns out they were very non-random, especially if we compared it to intellectual disability. And this is just showing one module called M3 that was enriched in chromatin modifying genes. In fact, the two major developmental modules expressed at the peak of cortical neurogenesis, which is between 13 and 20 weeks gestation, all were either enriched in transcription factors or chromatin modifiers, probably involved in key aspects of pattern formation and neurogenesis and fate, cell fate, et cetera, and morphology and differentiation. And so this was pretty remarkable, especially when we compared it to intellectual disability, which showed remarkably less specificity at the time, given the same sample size. Now I could say that the genes that affect intellectual disability, because there are many more that have been identified through exome sequencing, actually have some overlap with these pathways as well, but it's not complete. So what we had at the time was that if we compared rare de novo variants in autism to those that were differentially expressed in postmortem brain, to candidate genes and GWAS hits that were subthreshold, compared that to intellectual disability, and then asked about molecular networks, in other words, co-expression networks, genes that were highly co-expressed during brain development, we essentially saw kind of two major patterns. And it's just shown here on the right that you would have, and figure birth is kind of occurring right in the middle of this graph showing transcriptional regulation and synaptic, so birth would be there, that you have these genes that are de novo, large effect size mutations as shown in the M3 at the left are expressed early at the peak of neurogenesis, and they go down to the point where they're very lowly expressed in postnatal brain as a group. And then synaptic genes and signaling genes kind of go up. They're expressed during mid-gestation, but they continue to go up as synaptic development proceeds over birth. And so what we have there is we saw that autism genes converge to disrupt what we call neural development and cortical connectivity. Why do we say that? Because at the time, there was a enrichment of risk genes in these superficial cortical layers, layers 4 and 2, 3, which are the layers that connect different cortical regions to each other and connect the two hemispheres. Some of the genes do impact layer 5 and 6, which project down out of the cortex. They definitely do, but it's less of an enrichment than for the superficial genes. What was interesting about that from a molecular standpoint is that that also overlaps with imaging findings that showed disruption in long-range connectivity. But this kind of beginning look at this, and when you look at the network on the left, we don't understand what most of these genes do during brain development. Many of them have been identified in cancer or in other cell types and have some known function, but it really led us to this following thought. And let me just emphasize again that the genes, the de novo mutations in red are occurring in these transcriptional regulators, and the synaptic development genes are more likely to be those impacted by lower effect size variants. So it led us to kind of thinking in this way. Again, if you think from an evolutionary and population genetics standpoint, de novo mutations are new and they have very large effects, so they haven't been removed by natural selection, whereas things that are shared in the population are ancient and common. Those are the blue things. And if we kind of overlap those onto the two main functions that we saw disrupted, we see that transcriptional regulation are the de novos, and synaptic development function are more enriched in the common. And if we kind of now ask, where does this fit in terms of what's known about transcriptional regulation and transcriptional regulatory networks? There's a sense of a kind of tiered networks that are either 3 or 4 tiers high, with the top tier regulators being transcription factors and major, whose effects then are on the expression or splicing of other genes. So the top tier regulators are those kind of regulatory genes that have widespread effects, and so it fits in a way. This genetic architecture that we're seeing parallels what you might predict from a molecular regulatory architecture, in that large effect size mutations are more likely, not restricted to, but more likely to be in genes that have large scale effects that are at the top tier, because those effects kind of percolate through the network. But this really highlighted to us early in 2012 and 13 the need for having a refined understanding of regulatory networks in brain development if we really wanted to understand what these genes were doing. Now, I cite a review here. There's quite a bit of work going on in this, in this area. It's not as if there was nothing known, but really, if we compare it to other lineage studies, like in hematopoietic system, we're really far from that. We really needed to move into that area. And another thing that really kind of hit that we recognized at the time is that most of the GWAS hits were sitting in non-coding areas. And so if we wanted to understand how they were acting and what genes they were acting, we had to better annotate those non-coding areas with respect to brain, especially because biology is driven by gene regulation that occurs at the cell type and tissue level. The notion that it's not just what's going to be open, what's going to be active, what transcription factors are expressed, all of those things go into how a gene is going to be turned on. And a lot of that is cell type, not tissue specific or stage specific. And then we just heard, I got on at the end because I had something else going on and I apologize, but about activity dependence. And of course, that's a whole nother dimension that's extraordinarily important for the nervous system. So that's another, that's like, let's call that the 5th dimension. And so, recognizing that, we realized there was really very, very little known about, especially about developing human brain. And this is summarized in this title of this wonderful movie. If you like science fiction, on William Gibson that my wife and I helped produce a long time ago when our 20-year-old was 6 months old, because he's in the movie. But this title, No Maps for These Territories, which speaks about the future, really resonated with me as I was thinking in 2013 and 14 about kind of what we needed to do. And I wasn't sure what tools there were until Hae-Jung really came to the lab. Another set of, there were a bunch of papers popping out too, identifying, pathways that were affected in psychiatric disorders and implicating histone and chromatin remodeling as well. So there was, this other kind of, a lot of literature pointing to this. So, and we had started a study in 2011 that took a long time with, they all do when we're studying human brain, a histone acetylone-wide study of autism, taking 2 cortical regions and cerebellum with Sham Prabhakar at GIS, looking at H3K27 acetylation patterns. And again, we showed, there was a shared kind of chromatin enhancer pattern disrupted in autism that actually corresponded quite well to the transcriptome dysregulation that we had identified before. So again, just a lot of evidence telling us, boy, we need to look in this area and we need to make better maps. So what Hae-Jung started with was to map chromosome conformation using Hi-C during human brain development and understand its relationship to gene regulation. And we started this soon after the first Hi-C papers had come out in 2013, Job Decker, Bing Ren, Lieberman, and then Brode. And then the idea was to leverage Lieberman, Aiden, leverage the chromosome conformation data to annotate non-coding regulatory elements and common risk variants for schizophrenia and other disorders. And this again was led by Hae-Jung Won, and some of the other work I'll discuss was also with Jason Stein and Luis de la Torre Ubieda. So again, we're generating a reference map of gene regulation in fetal human cortex. And the idea was, we started with a very simplistic notion, but we wanted to have some known biology as a guide for us. So what's shown there on the right are the laminations. It's just a DAPI stain of the cortex. And we have the germinal zones on the bottom where neurons are being born. Then they migrate up into the cortical plate through the intermediate zone and subplate. And that area, cortical plate, expands to become the adult cerebral cortex. Or, certainly all those neurons are born before 26 weeks in gestation, again, with a peak kind of between 13 and 18 weeks. So we wanted to know what genes are expressed, what regions of the genome are active, what genes they regulate, and how do they regulate them. And then I cite some of the papers that have started to integrate these data below. But I'm going to start with the Hi-C data. So this is what Hae-Jung did. I'm not going to go over this in detail. This was again done around 2014-15. You can see we used a pipeline, kind of what I would call a standard pipeline at that time, and Hae-Jung called loops by fitting a Weibull distribution, which looked appropriate at the time. So the first thing that Hae-Jung did was first, we looked at the other data that was out there. There weren't that many papers yet in other cell lines, papers from Bing Ren and from Job Decker's lab, and we had kind of, we were able to identify, compartments and TADs, and there was a high overlap between our data, our TAD calling and theirs, et cetera. So that made us feel that we had decent data, about a 0.8 correlation or so. What we're really interested in is asking, are these Hi-C loops that we're defining functional? Because the TADs were largely, not 100%, largely preserved over cells. So how do you get cell type gene regulation that's specific? And so we reasoned that's going to be in the looping, which is bringing enhancers and promoters into contact. And so these maps were made at a 10 kb resolution from a set of n equals 3 for each germinal zone and cortical plate region. And so then Hae-Jung with Arima-Hic had looked at the relationship between eRNA and mRNA, and we could see that there was a strong correspondence between eRNA, mRNA correlations and loops. We did not have fetal brain EQTL, it was adult brain, but we still asked, are genes that are physically interacting here with high C at the loop level with EQTL, are they significantly associated with EQTL-associated transcripts? The answer was yes, and also used DHS openness promoter correlation and showed a kind of very similar thing. And I'm just going to give a very, very quick example of one of these just to show you what I mean. So the idea is that if a high C interaction between a variant, let's call it, the T and C sitting in the middle, is actually a regulatory interaction, that it should be non-randomly associated with expression quantitative trait loci. In other words, with the gene expression. So, is T associated with gene B, A, or C? And so what we were able to do is basically ask at the overlap. We had this EQTL data set from Ramaswamy et al., which was a underpowered but nice data set from human brain with a few, about 150 samples, maybe 120. And so it's of course an incomplete map, and so was our high C map, by the way, because we don't have a huge number of samples. But we could see that if we look both at the germinal zone high C libraries and the cortical plate libraries, that we had very high overlap, quite significant overlap between, the EQTL. It's about a 30-something% overlap between the high C peaks and the EQTL identified. This is highly significant. So it gave us some evidence that what we were looking at were these loops were likely to be en masse regulatory. And so of course, then we went to ask, can we use this to annotate GWAS signals? And Hae-Jung chose schizophrenia because there had been this beautiful paper in 2014 from PGC that was a turning point in the field by identifying 108 schizophrenia-associated genetic loci. But in only 10 instances was the association signal really clearly attributable to a change within a known gene. And the way that things were done was as follows. You basically look along the genome and you assign SNPs, the index SNPs identified in a GWAS, to the nearest gene. And or you could take all genes within the linkage disequilibrium block, the LD block around that SNP, and say, okay, these are the candidate genes. But of course, in some blocks, there might be dozens of genes. So there's still quite a bit of work to be done. And you could do overlap with EQTL, which is a step better, but that wasn't done in this case in that 2014 paper. And there frankly wasn't much EQTL data in brain yet to speak of. And we decided, boy, if these loops are defining functional enhancer promoter interactions, then we could actually assign genes to SNPs to genes using the loops. And another reason why we thought this would be a good idea is because in looking at the known EQTL data, most of which was out of brain, we could see that at least half of EQTL were quite distal. In other words, they were not acting on the closest gene. So it gave us a really substantial reason to think that this would be a better way to go. So we saw, based on the high C data, that only half of the GWAS loci could be mapped to the closest gene using this. And many are acting more than several 100,000 base pairs away, 95% within the same tag, but still that could be hundreds of thousands of base pairs away. This is an example of one locus. The credible SNP is identified there, the GWAS locus, and its credible SNP. We used a mapping technique called Caviar, bioinformatic technique. And so there's the GWAS locus with the LD block in pink. Then below it, we have the TADs from the GZ and CP labeled in different colors. And then the negative log 10 p-value of the high C interactions, one there, and a very, very large one there, especially present in cortical plate, which is post-mitotic neurons. And so we look here, we see these SNPs, and we see that what it's interacting with is something entirely outside of the LD block and appears to be this acetylcholine receptor promoter. So we identified by assigning this to the right gene, we were actually able to implicate new pathways, including cortical neurogenesis and acetylcholine receptor activity. There were numerous cholinergic receptors that were identified in this way. And we were also able to validate that quite a few of the SNPs, about half of them, were actually interacting with the closest gene in some cases. But you couldn't a priori know that. This is another locus that's just far outside of the LD block and just showing what that looks like. So, again, we saw numerous cases like this. And so what we were able to do is now use CRISPR in primary neural progenitor lines to take out the region surrounding these loci. I'm going to show a different locus that's FOXG1. It's another gene involved in proliferation like SOX2 is, but it was just almost 700 kb away. So it was just so interesting. There's the SNP in between PRKD1 and this non-coding RNA, and there's FOXG1, and one could see that this prediction from the Hi-C map would be that the region that SNP is contained within, which is a 10 kb region again, let me be clear about that, interacts with the FOXG1, which is hundreds of thousands of base pairs away. And so we made a few different snippers with CRISPR and showed that we were able to, that it had a specific effect on the FOXG1 and didn't have much of an effect on the closest gene. And we did this for a number of genes, used some other methods to validate some of these as well. So this, and this is a kind of a nice form of biological validation. Subsequent to that, we've kind of incorporated in the same, using the same model, the germinal zone and cortical plate, with Attack-seq maps to look directly at chromatin accessibility, which has the advantage that it's also kind of a smaller region. Our average Attack-seq is about 1,100 base pairs. And we reasoned that if we could take open chromatin that's in a promoter and look within a TAD and find the most correlated in terms of read pileup open chromatin regions, those would likely, be the likely enhancers. And indeed, that's what we saw by overlapping this type of data with Hi-C. We think we got the highest confidence kind of regulatory interaction. So we use this to define a higher resolution map of non-coding regulatory elements active during neurogenesis. And we studied, we did a bunch of studies with it, including re-annotating GWAS. And I'm not going to go into the details here. It's published, but I just wanted to make the point that these data need to be, are optimally used when they're integrated with other data. And more recently, defining EQTL in fetal, we finally, after years, made an EQTL in fetal brain. It took us years to collect enough brains. We had 220 brains, and this was led, again, by Jason Stein when he was in the lab, and Luis de la Torre Vieta, but really taken to completion by Rebecca Walker, a graduate student in the lab who's now finished. But the idea was we had over 200 fetal brains, and so that was a reasonable size. We identified about 6,500 E genes, and we were able to identify splice QTLs as well. And using single cell sequencing, kind of deconvolute some of this to a first approximation to cell type specific patterns. And so we integrated this with looking and asking, where, using LD score regression, where does the heritability for various, disorders sit? We used this for a transcription-wide association study, and we conducted co-expression network analysis to identify modules of co-expressed genes that might be enriched in various disease variants. But one of the points I want to make is, again, just like before, if we compared a similar size data set from adult cerebral cortex, we can see that we're identifying a similar number of E genes, but the overlap is not even 50% in a similar size study. Again, the point being that one needs tissue and stage specific maps here. So one of the things that Rebecca also looked at, like we had before, is asking if we look at the presumed EQTL sitting within enhancers, acting to increase or decrease transcription. We asked about their overlap with attack-seek correlations and high-C loops. And so if we take all EQTLs, both distal and proximal, distal being further than 10 kb away from the EQTL peak, we could see that a large percentage, more than half, are supported by the attack-seek data. In other words, the correlation of enhancers and promoters. Now, for high C, because we only had 10 kb resolution, we could see that it was lower, but it was still quite significant. About 25% of the distal EQTLs overlapped with a high C loop. Now, why is that? It could be that obviously we've only done a total of 12 high C experiments from 3 different brains. So we probably need a much larger and kind of deeper sequenced cohorts to have a more complete high C looping map. But in any case, I'm happy to discuss that. So just to conclude this, and then do I have 5 minutes left to just show a few Arima things? Is that okay?
Sure, definitely.
Yeah, so I just wanted to show you kind of where we had been going, and I just summarized that here. That we're able to validate these data, that they're really, really powerful, we think, for connecting loci to genes, enhancers to genes, etc. And we think that integrating with other data may improve the predictive ability. And this is just showing several of the papers where we've made, I'll be honest, relatively minor contributions by basically integrating the high-C and attack-seek data, these functional genomic data, with GWAS data to actually do a better job of annotating actually what are the likely candidate targets of these GWAS loci. And this, I think, was culminated in our PsycEncode project papers, both with Schahram Akbarian and then the Wang et al. With Mark Gerstein, where we integrated adult high C data with other data and really showed how it can increase the power of machine learning, deep learning methods to kind of identify new candidate genes. And all of this kind of culminated in Hye-Jung. She had started this when she was in my lab, but it was her idea and definitely not my idea. This is essentially what we were doing. We were using these tools to assign these functional genomics data to assign SNPs to genes, and so she attached it to Magma, which we had been kind of doing in the lab, but not in a formal way, to create a tool that now allows you, instead of having the genes assigned to the closest gene or the LD block, to actually use these functional genomic data to improve the prediction of risk genes. So I urge you to go take a look at that if you're interested in this. The rest of the work, which has been going on for a couple of years now, is work from Jenny Yin, who came to the lab, and she's doing she's really taken over where Hye-Jung left off and is exploring the impact of structural variation, as well as looking in postmortem autism brain. She has multiple projects, but I just want to discuss the structural variation stuff because it's a kind of another spin on this. Here's the idea. We have a disorder like 15q duplication, which causes about 0.6% of autism. It's highly penetrant for autism. And there's a big region with multiple breakpoints that is duplicated in this syndrome. And one of our notions was, we always think of these CNVs as just affecting the genes within the region, but how about the distal effects? How about effects on gene expression that are resulting from epigenetic changes in TAD boundaries or looping or compartments, et cetera. And so when she started this, rather than just jumping on our ongoing high C, which was working, but quite expensive and needed a lot of tissue, she compared these 3 methods, and she's presented this before, so I'm not going to go into gory detail, but leave me to say that we're able to use the Arima-HiC, probably because of the cocktail approach and some other things, to get really good high-C maps from almost a tenth of the number of cells. So it's definitely been an improvement for us. This is data that Jenny's presented before, but I just want to, one point here was that the easy high-C in our hands did not work very well. That could be because those are our hands, but compared to the Arima-HiC and our regular in situ high-C, the you know, the original high-C from Lieberman. But you know, we're getting quite good maps. I'm not going to go through all the technical details, but I'm happy to in questions. But one of the points here is that we did this in sorted neurons that were done, given to, where Jenny sorted them with new N positive and new N negative using a protocol that Schahram Akbarian had given us, and that really works quite well. And so you can have the new N positive on the left, and the new N negative on the right, the controls on top, the 15q duplication on the bottom, and we can see that within cell there's a very high correlation between the high-C matrices. I think it's at 100K resolution. Jenny can correct me, but some, you know, it's not a, this is 100K, I think, or something like that. And the same, you know, and you can see again within cell type better than between the cell types, which is kind of what we expected. But good correspondence actually between the maps at this resolution, which is what we expected. And essentially, Jenny went through looking at many methods, and she could discuss her choice. But she did an A/B compartment analysis, a TAD analysis, and of course the looping analysis, just like Hye-Jung had done before. But we're using slightly different methods now. There are a lot more methods on the market, right? And this is one of the major findings to date, is that if we look at the A/B compartments, it's not just in this brain region. We actually can look in other brain regions and see this as well. That within the new N positive cells, we see a big compartment switch inside and around the duplication, but we don't see that in new N negative cells. So this compartment switch is cell type specific. And Jenny is now working on getting deeper libraries and looking at looping and TAD boundaries and all that. I'm not going to, because of time, I didn't want to go into that, but just show you the main point here was, looks like there is some cell type specificity to this compartment change. And she's done this in I think it's 6 cases in control. So this is not just one case. A second structural variation that Jenny is working on is this famous chromosome 17 inversion that's about 900 kb that leads to a tau haplotype, H1 and H2 it's called. It has a different frequency across different populations, but the H1 haplotype is of interest to us and many others because it is a major risk factor for neurodegenerative diseases, including progressive supranuclear palsy, which is a Parkinson's plus syndrome, in which about 96% of the patients have this H1 haplotype. The tau gene is circled there in the middle. There are probably other things involved. And so our hypothesis is that the gross structural differences between haplotypes will confer effects on the regulation of gene expression in this region, as well as the wider transcriptome. And so this is work done with Alison Goat at Mount Sinai, where in her lab, her postdoc has been growing, and these were made by Celeste Karch originally from patients who were homozygous for H1 or H2 haplotypes. And then they're grown into excitatory neurons, astrocytes, or microglia. I'm just going to be talking about the excitatory neuron work right now on the high-C chromatin structure. But one of the really interesting things that Jenny kind of solved is that we realized we had to call loops properly. We had to construct reference genomes of the 2 haplotypes separately. Because if you look to the right, I mean, one uses the distribution of contacts with relevance to their genomic distance to kind of call loops. And if you've misspecified the genomic distance and that it actually is much closer or much further, you've changed your denominator. you're using the wrong denominator. I hope that makes sense. So within this inversion, and so we mapped each onto its respective actual true structure, which took quite a while to do. She used 2 TAD boundary calling programs. She can talk about why she chose them, top dome and looking at the insulation score. And the top dome are blue, the insulation score is showing the likely TADs in the dark blue below. She did an N of 3 of each, and so the H1s are shown up above and the H2s below, and one can see that there are quite a few conserved TAD boundaries, especially outside the region, but inside the region we can begin to see changes. And in fact, that's shown here, showing both using the insulation score and the top dome. We can see that there's, if we go between the H1 and H2 and ask for, what are the percentage of TAD boundaries that are shifting, we see, it's pretty low, close to 0, and then it pops up, within this region, which is that huge peak, and then both the insulation score and the top dome show the same thing. So that's telling, that's pretty interesting, and telling us that what we're seeing in this locus is not a artifact of the way we're doing the analysis. It's actually happening within this locus. And so then, Jenny's done a lot more work, but I just, again, wanted to summarize one thing here. This is just showing the consistent loops between the that is those that are seen in more than one of the samples. And there are many consistent loops that are, that are still there, and the loops are shown in the dark kind of purple. But one can see clearly there have been huge shifts in the TAD boundaries here. And so she can look to see if there are haplotype specific loops. And here are a few that have been identified. And she's now characterizing them in much more detail. But again, this just shows how the kind of Hi-C method can be used to look at structural variation and its effect, to begin to understand the mechanisms that affect gene regulation. So the rearrangements between the H1 and H2 haplotypes affect both TAD and loop structures. And we think that they affect many genes within the locus by linking the genes with different enhancers or different methylated CG dinucleotides. Some of the areas that are being linked aren't kind of known enhancers, but are CPG rich. So anyway, thanks for your attention. I thanked everybody up front, and I apologize for going a few minutes over. I just wanted to show some of the new stuff that we're doing and some of the mess that we're in. So anyway, thanks.
Thanks a lot, Dan. So maybe it's time for 1 or 2 questions here regarding the first part of the talk, and the utility of integrating chromatin interaction data like Hi-C with other features like open chromatin for linking disease-associated variant regions to target genes. You showed something there about the CRISPR-based validation, and one of the things that commonly I think comes up when when I also speak about the utility of this data for that purpose is, how do you feel like you need to validate using CRISPR, and if so, how to sort of scale that process up? Yeah. So I do feel that, especially if we want to make a biological point and really prove that the region is being regulated, we do 2 things usually. And I didn't, we did, there's a lot more validation in all those papers than I showed you. I just skimmed over one example for each, but I'd have to say that when we pick high confidence interactions, that is those that overlap with Hi-C and ATAC-seq, I'm fairly, we get, we haven't done hundreds, so I can't give you a number, but really a lot, most of them are validated.
That's number one. When we pick things that, you know, where we have an ATAC-seq enhancer promoter correlation and a Hi-C loop, those have been readily confirmed with CRISPR in the right cell type. Surprisingly so. And in fact, even what Haejung tried, most of hers validated even without the ATAC-seq, the majority. But again, I don't think we did enough to be 100%, you know, to put a number on that. We also do reporter assays as well. So we ran some reporter assays just to show that the variant itself was actually changing expression, you know, the SNP that we were looking at. But to kind of validate the Hi-C, you know, I think there are a lot of methods out there now that we've been toying with, but we're not expert in yet, which involve kind of multiplex CRISPR libraries to, and then doing single cell sequencing to recover the barcodes so that you can actually identify the cell type as well as the gene expression changes in that cell type. And so we've started to do some of those pooled CRISPR screens. I think if your goal is to test thousands rather than a few dozen, that's a way to go. And it also gives you the cell type resolution. You know, there are others who are much more methodologically advanced than we are, who have been doing this kind of thing for a while. So we've started to do some, you know, some of these pooled CRISPR screens. You know, we're doing that when we have hundreds to, you know, high hundreds to thousands. I think for a couple dozen, the CRISPR is easy enough to, you don't need to do anything fancy. I think just choosing the right cell type, primary cells being better than cell lines, because that's one of the points here, is that a lot of these things are stage specific. But does that make sense?
Yeah. Yep. Okay. Well, I think given the time, maybe we'll pause the questions for now, and I can hand it back over to Joe to wrap up. And thank you very much, Dan, for your excellent talk.
Oh, wonderful. Thank you, Anthony. And big thanks again to the speakers, Schahram, Jon, and Dan. Thanks to the attendees for joining us. Quick promo, visit our Twitter account for latest or website for resources, links to pubs, testimonials, videos on our YouTube. Thank you, everybody, and have a great day.